Before I introduce Professor Pat Chow Fraser from McMaster University, I wanted to show you this photograph taken by the Water Brothers with their drone 2 summers ago to show you the kind of work that is been undertaken by Professor Pat Chow Fraser's lab to understand what's happening in these high quality, diverse wetlands. This is a photograph of doctoral candidate Kelton Adderley Heron tracking species at risk turtle movements. They have put tracking devices on turtles and on regularly have been tracking their movements and keeping a record of where they have moved. So I will let Professor Pat Chow Fraser get into the details of this research, but I thought this would be helpful for you to see what's involved in doing this work. I want to explain to you that when we first started working on this, our hydraulic engineers figured out that there was likely erosion happening in the St. Clair River. So way back in 2003, with GBA Foundation funding, we retained W.F. Baird and Associates, coastal consulting engineering firm, internationally respected. They determined that erosion was happening in the St. Clair River. We took those findings to the International Joint Commission. They undertook the International Upper Great Lakes Study. We were pleased with that because we thought they could probably carry on that work, figure out what was happening in the St. Clair River and what could be done to prevent further erosion from happening. So we hosted the study chairs on Georgian Bay at one of the dried up wetlands. And Dr. Carl Schiefer accompanied us on that visit to that same dried up wetland near Cognachine. And the US study chair turned to me and said, Mary, you tell me the top 10 wetlands in Georgian Bay, and we can come in and dike them at a cost of $1 million each. And that will cost a lot less money and a lot less grief than fixing the problem in the St. Clair River. I can tell you that I was stunned at his remarks. Carl Schiefer was so stunned, I wasn't sure if he was going to pass out. I said to him, what will that do for the fish? Oh, he said, we will build a fish ladder. And Carl responded with, these are bass, pike, and muskie. They cannot move up and down fish ladders. Well, he said, then we'll just set traps and put them in here. And I said, what will happen to this wetland, this dried up wetland that you will have flooded? What will happen to the fish in the fall when this freezes? And he said, oh, we will come in here, we'll trap them and put them back out in open Georgian Bay waters. That would do nothing for the rest of the aquatic life, the snakes, the turtles, all the little tiny minnows that would be in that wetland. We know that diked wetlands don't come anywhere close to matching the ecological values of an open wetland. But besides that, of the hundreds, if not thousands of Georgian Bay wetlands, it would be egregious to even try to figure out what 10 wetlands should be diked. I think he realized how stunned we were, but that's where that study started, and that's where it ended with a do nothing recommendation. Today, here we are today with nothing having happened, but fortunately the IJC commissioners rejected that study findings and recommended that bathymetric measurements be taken of the St. Clair River and methods to be able to control the outflow during low supplies, but that those flexible measures not be deployed during high supply conditions. So here we are today, 20 years later. The water levels are declining after record high water levels. We want to finally figure out a solution to this. So to help us understand what the impacts are of those extreme levels, I'm introducing now Professor Pat Chow Fraser of McMaster University. She and her graduate students have been conducting research for 20 years on this work, and no government agency has ever undertaken this level of ecosystem research for Georgian Bay sensitive Canadian Shield waters and wetlands ever. So with no further ado, I'm going to introduce Pat Chow Fraser. Thank you.
On behalf of my postdoc Prabha and my students Jonah and Jacqui, I'm Pat Chow Fraser of McMaster University, and I welcome you to a presentation on the health status of coastal marshes, at-risk turtle habitat, and the nearshore water quality of eastern Georgian Bay. So the projects that are going to be covered in this talk involve the wetland vegetation changes in coastal marshes of Georgian Bay under contrasting water levels, habitat use by blandingii turtles in the Tadenac Bay area, and the long-term changes in phosphorus and E. coli in nearshore waters of Georgian Bay Township. So the first one that I'm going to present on behalf of Prabha is on wetland vegetation changes in coastal marshes of Georgian Bay under contrasting water levels. Overall, the coastal marshes of Georgian Bay are considered quite unique within the Great Lakes Basin. The shoreline of Lakes Erie and Ontario are sedimentary that include things like shale, limestone, and sandstone that are easily eroded. And this allows for accumulation of a lot of sediment and for extensive cattail marshes to form along the shoreline. And these marshes are typically can migrate up and down the shoreline with different water levels. In eastern and northern Georgian Bay, on the other hand, wetlands have this granite bedrock that do not weather well. And therefore there's only a thin layer of soil that is present in these wetlands even after thousands of years. These conditions don't allow for formation of these large cattail marshes such as those in southern Ontario. Another difference is that many of the coastal marshes in Lakes Erie and Ontario have already been destroyed or degraded, but those in eastern and northern Georgian Bay have only had limited development pressure. Most of the areas were logged in the 1800s, but there's been secondary growth of forests since the beginning of the 1900s. So currently, human impact has been limited to recreational development, which is quite a bit different than the intense urbanization and agricultural development on the shores of Lakes Erie and Ontario. Now, this graph shows average deviations of Lake Huron water levels from the long-term mean that dates back to the 1918. The blue corresponds to above average levels, while the pink correspond to below average. The graph shows here the mean water levels between 1990 and 2015. And the graph clearly shows that there were 14 continuous years of low water levels below the long-term mean, beginning in 1999, and that ended in 2014 when water levels started to climb above the long-term mean and then eventually hit the record high levels in 2020. This pattern of water level fluctuations have not been observed in Georgian Bay or anywhere else in the Great Lakes over the past 100 years. So why does this change in water level matter? It matters because different types of plants are adapted to living in different water levels or depth of the water table. So for instance, most trees are quite intolerant of any length of inundation. The meadow species will tolerate some seasonal inundation. Emergent plants like cattails have a range of tolerance of water depths, and all of the floating and submergent species have to occur in or on water. So the water of the Great Lakes, in this case Lake Huron, is going to fluctuate annually. The water's edge essentially separates the ecosystem at any point into a terrestrial part that occurs above the shoreline and an aquatic part that occurs exclusively below the shoreline. This has also been termed the high marsh, above the water, and the low marsh, which is below the water. The plant species that live in the terrestrial zone include trees. There are shrubs, sweetgale, sedges in the meadow zone. And then in the emergent, which kind of straddles the terrestrial and the aquatic, we find the emergent plants like cattails and bulrushes. And then within the water column, on top you have the water lilies, and within the water column you have the pondweed. Now during the normal cycling of highs and lows, there will be no net change in the high marsh or the low marsh. And that's because during periods of high water levels, which last typically in the range of 3 to 7 years, the emergent vegetation will either die back or expand. But because these lengths of inundation and drawdown do not last more than a decade, the net change will be zero. But during the period of sustained low water levels that we experienced, such as between 1999 and 2014, there was a net expansion of terrestrial habitat at the expense of the aquatic habitat. So some of the trees started to move into the meadow zone, and the species of meadow plants moved into the emergent zone, and the water lilies and the submergent species became trapped in that condensed water, very, very shallow. And then from 2015 to 2021, the meadow zone became flooded when water levels rebounded to record high levels. Now, in the previous century, whenever water levels stayed below the norm for 5 to 7 years, the meadow species that became established would be eradicated by the high water levels. And in fact, the literature predicted that within 3 years of high water levels, the coastal marshes in Georgian Bay would return to conditions experienced prior to 2000. Nevertheless, the prolonged drawdown in the early part of the 2000s had allowed pine and alder trees to become established in the meadow zone, and for the shrubs and sedges to establish in the emergent zone. So when water levels rose in 2015 and beyond, the trees began to die, but very slowly, and they remained in the meadow zone. And in some cases, the meadow species were able to persist and even thrived in the high water levels. And this precluded the reestablishment of the submergent and floating vegetation that would otherwise have accompanied the higher water levels. So although the water levels had been restored, the plant community and the habitat structure had not been restored. And this had in fact become a novel habitat that had not been seen in Georgian Bay or anywhere else along the Great Lakes coastline. So there has been a clear transformation of the vegetation community from one that's dominated by submergent and floating emergent vegetation that are clearly required by the fish community to this novel habitat of flooded meadows with many of these dead and dying trees, shrubs, and sedges in the aquatic portion of the marsh. Now, since this change in structure has implications for fish and wildlife, we knew that we had to conduct a proper study to not only document how the habitat has changed, but also to quantify what has changed. So there are several study objectives. The first, we had to quantify changes in the vegetation in various regions of eastern Georgian Bay. We knew that we could not just focus on one region because they are very, very different within the entire coast. We also wanted to determine the amount of this novel habitat, which included the dead trees. And then we wanted to relate these changes to the vulnerability of wetlands to water level disturbances that we had predicted by another student, another PhD student in my lab, Dan Weller. So to do this project, we relied on two main sources of satellite imagery. The first is the ICONOS images that were acquired by the Georgian Bay Foundation in 2002 and 2003. And that covers the entire shoreline of eastern Georgian Bay, shown here as the green and the red rectangles. These images captured conditions of wetlands following 4 or 5 years of extreme low water levels. We then also had to acquire archived satellite images produced by the satellite Pleiades. In 2019 was the year that we settled on. This was a year that we had a fairly good coverage of the eastern shoreline. As you can see on this map, we could not get all of the entire region that we had in 2002 and 2003, but most of it was included. And so we use these to quantify the habitat changes in coastal marshes, and then subsequently we'll be using that to assess how fish and other wildlife may be affected by the change. And to do this, we're going to be using remote sensing to conduct a change detection of the wetland vegetation between periods. Now, along with the satellite images, we also required ground reference data. So there were times in 2020 and 2021 where we went specifically into certain wetlands to collect data. We also accessed data that had been collected by other students for other previous projects. We also used images that had been acquired by a drone, an unmanned aerial vehicle, for other studies that we had ongoing. And then we also used some orthophotos from the Coop and Scoop series. Now, just to make this complete, I'm showing you the process that we followed as well as the tools that we used, which included ENVI, QGIS, as well as ArcGIS Pro. So the first one that we looked at was an area in Point Barrow that moderately high recreational development. Then there was a region along Franklin Island, which we also had substantive data collected in the field. And this is a region that we considered having moderate recreational development. And then there's Tadenac Bay area, which has mostly undisturbed coastal wetlands, and that's also managed and protected for fish and wildlife. And then finally, Severn Sound and Honey Harbor area, where we know has heavy recreational development and is a region that we have studied almost continuously since 2003. Now, as with most of the project where mapping is involved, we have to do a classification accuracy assessment. The overall accuracy refers to the percentage of all the pixels that are correctly classified. So this includes everything that's in that image. And generally, anything approaching 90% is excellent. And you can see here that except for 2019, for Tadenac, we had all of the other overall accuracies were above 90%. Now, because we're specifically interested in the dead trees novel habitat, we also needed to determine the user's accuracy and the producer's accuracy. So first of all, the user's accuracy represents the probability that a pixel classified as dead tree is actually dead tree on the ground. And the higher the user accuracy, the higher is our confidence that we have mapped the dead trees correctly. And so this is considered reflective of the commission error. On the other hand, producer's accuracy represents the probability that all of the dead tree pixels have been correctly classified. So this is different from the user's accuracy. The higher the producer's accuracy, the higher is our confidence that we have in fact mapped all of the dead trees that are present on the ground. So this is reflective of the omission error. So if we look at 97% user's accuracy and 92% producer's accuracy for Severn Sound, this means that we can be confident in having mapped all of the dead trees correctly and haven't omitted any of them in a substantive way. So we should start out by looking at a fairly high level comparison of the percent vegetation cover in 4 regions according to the habitat class. So I've split the data looking at each region separately according to each of the vegetation classes. So what you see here plotted are the mean percent aerial cover with 1 standard error bars. The blue bars are corresponding to the low water levels and the red are corresponding to the high water levels. And in the case of the dead trees, because we didn't have any of them during the low water levels, there are no blue bars for that class. And generally you can see that there's high variation among the different classes with respect to percent area, and that there are also substantive variation among the regions. And in fact, there is only the meadow, emergent, or shrub class where there were significant differences for all 4 regions with respect to the water level scenarios. And in this case, there were much lower of the meadow emergent vegetation following the high water levels. On the other hand, for the submergent vegetation class, we see that for 2 of the wetlands, namely the Portobello and Franklin Island, we had SAV that were much lower during the low water levels, whereas in the Severn Sound and Tadenac, they were much lower during the high water levels. So let's look at the within region variation in Tadenac Bay as an example. We had 4 wetlands in this region that we had sufficiently sampled between 2002 and 2019 for us to do this automated classification. And to see a little bit more detail, here I'm showing you David's Bay with the Iconos image, the Pleiades images, and then the classified images on to the right of them. Here you can see also Black Rock Bay and then wetland number 1. Now I've provided this comparison between Tadenac Bay and Franklin Bay to show you how the different regions responded to the high and low water levels. You can clearly see that within Tadenac Bay there were not as much of the dead trees compared to the Franklin Islands, the Francis Bay and the Corbin Bay. So in the Tadenac Bay there was more open water but significantly less submergent vegetation, probably due to inappropriate water depth, and this is because of the wetland bathymetry. With the Franklin Island, we had loss of meadow and emergent vegetation in favor of submerged aquatic habitat, but we also saw an increase in dead trees. As our last objective, we wanted to relate the percentage of the novel dead tree class to a vulnerability index that was developed by Dan Weller, former PhD student in my lab. This vulnerability index, or the VI, is based on wetland bathymetry and exposure. Generally, the more gradual the slope of the wetland, the more vulnerable the wetland is to water level disturbances. Thinking of it in another way, the steeper the incline, the less likely the wetland vegetation will change along the shoreline. In this map, you can see that the higher VI scores have a red-orange color, while the lower VI scores have a light or dark green color. And Severn Sound is a region with relatively high VI scores, as indicated in this map with the hexagons in Severn Sound. When we regressed the percentage of dead trees in the 4 regions against corresponding VI scores, we found a highly significant positive relationship. And this positive relationship is confirmed by the 2 classified maps of Seven Sound. The top map here is of the 2002 ICONOS image. In here, you see no dead trees during the low water levels, but with areas of submergent and meadow vegetation. But then by 2019, in the bottom map, you see that during high water levels, there is a clear rim of dead trees all around that shoreline, and the percentage of submerged vegetation had been reduced, as well as the amount of the meadow. So it is very clear that the nature of the habitat has changed, and this is going to require more than a simple mapping exercise to determine what an amount has changed because of the creation of this novel habitat. We need to have more research to determine how fish and wildlife are using this habitat class, and we need to collect more data for site-specific changes, because we feel that it's very dangerous to extrapolate information from just 1 or 2 sites to all of the other regions of Eastern Georgian Bay because of the high variability in both exposure and wetland bathymetry. I want to thank the financial contributions from Georgian Bay Great Lakes Foundation, who partnered with MyTax to fund the postdoctoral salary. I want to also acknowledge the logistical support, in particular provided by Mary Muter and David Flower, especially in the Honey Harbor area, and all the present and past PCF lab members, but especially Danny, Nick, Rita, Kelton, Jonah, and Sherry. Thank you. So the next project that I want to report on is that carried out by Jonah Lehman as part of his M.Sc. and is entitled Habitat Used by the Blanding's Turtle in the Tadenac Bay region. We'll start by introducing the Blanding's turtle, which is one of 8 freshwater species in Ontario. It is known for having a large home range because they use many habitats, especially wetlands. And unfortunately, their numbers have declined because of habitat loss, road mortality, and other human disturbances. And they've been listed provincially as threatened and federally as being endangered. While there are many conservation initiatives involving blandings, they often occur in southern Ontario, where landscapes are highly urbanized and degraded. And if you look at the large number of green squares in this map along Georgian Bay, you would expect relatively good populations of Blanding's turtles in the Biosphere Reserve. Since the landscape in Eastern Georgian Bay is not like any of the developed areas in southern Ontario, where a large number of the published studies have been carried out, then we feel that there is an urgent need to conduct studies specifically for Eastern Georgian Bay so that we can put together recovery strategies for those populations living in Eastern Georgian Bay. Like many other undisturbed regions of Georgian Bay, the Tadenac Bay region, indicated in red, has more than 300 coastal marshes, hundreds more bogs and swamps and forests. The Tadenac Club owns a large portion of this region, which consists of 6,000 acres, which they have managed for over the past century as a fish and wildlife sanctuary. This area is known to support many species of freshwater turtles and other reptiles. But aside from a few turtle surveys, no proper research project has been conducted in this region to determine the movement and habitat use of blandings or any other species. During the 2021 field season, 8 Blandings turtles, 6 males and 2 females, were caught on the Tadenac Club property. The turtles were trapped in hoop nets baited with sardines, or they were caught opportunistically by hand. Upon capture, we glued a small radio transmitter on the shell of each turtle, and this allowed us to track their movements using an antenna and a receiver that can detect the unique frequency of each transmitter on the turtle. Blandings turtles were tracked in this way weekly throughout the summer, and the GPS coordinates were taken at each location and then entered into a GIS. Shown here in this slide are photos of some of the available habitat types for Blandings. These include the peatlands, some are bogs, the ponds with some shallow water. There are carnivorous forests. There are thickets, swamps, rocky outcrops, many inland marshes, as well as a fair number of coastal marshes and a deep water lake. This image shows you the relationship between the various types of habitats used by one turtle that we tracked last summer. For the turtle, it did not have to go very far to reach, for instance, the rocky outcrop, the deep water lake, the inland marshes, the coastal marsh, the peatland, the upland forest, or the thicket swamps, as well as using a large part of the shallow water in a pond between wetlands. During the summer of 2021, the turtles made use of their resident wetland, which is the one in which they were originally caught. The resident wetland was relatively large, extending more than 1 kilometer across within the Tadenac property. All 6 turtles spent majority of their time in the resident wetland, except for one individual, number 4, who spent majority of its time traveling across multiple wetland complexes throughout Tadenac. In June and July, 3 males made extensive forays outside to coastal marshes on the edge of the study site, each turtle remaining in these marshes for at least 1 week and up to 3. One noteworthy observation we made is a male apparently swimming across deep open water and continuing to travel for more than 7 kilometers from wetland to wetland during a single season. It was also observed crossing a busy road. And while Blandings are known to be making extensive overland movements, they are generally not considered good swimmers. And this shows that there are additional threats to this population, including the potential for boat injuries and road mortality, even though they are living in a wildlife sanctuary. The type of coastal wetland habitat used, as well as the length of time spent in these wetlands, differ from the population that currently reside in northern Beausoleil Island near Honey Harbor that was tracked by Chantal Markel in 2011 and 2012, which is the only documented study of Georgian Bay. While the 3 male turtles made use of dead trees and dead shrubs in coastal marshes in Tadenac, only one of the males in Beausoleil made use of marshes along the coast during the summer. We want to emphasize that hundreds more data points need to be collected before we can begin to determine habitat use. Therefore, the 73 data points that we have collected so far cannot be seen as anything but preliminary. Nevertheless, it is interesting to note the extent to which these 8 individuals used shallow pond water throughout the summer. And as noted before, they also appeared to use coastal marshes much more than had been documented for the Georgian Bay Island National Park population. So it appears that the primary habitats used by Blandings turtles in Tadenac Bay differ markedly from those on the Beausoleil Island, where they depended on vernal pools and bogs. And this highlights the importance of conducting more studies in eastern Georgian Bay rather than extrapolating from published studies. In February 2022, Jonah was able to go up to Tadenac with assistance provided by GBGLF and the Tadenac Club. He was able to locate the overwintering sites of many of the turtles, which are shown in this map. These locations were triangulated by using the frequencies transmitted by the radio tags that were left on the turtles for this specific purpose. 3 of the turtles overwintered in the same marsh habitat that they were found in, that we call the resident wetland. 2 overwintered in separate thicket swamps. 1 overwintered in shallow water habitat, and 1 overwintered in a marsh habitat on its own. If we compare these results to published findings, blandings are known to overwinter in marshes and ponds throughout their entire range. This contrasts those of Marco and Chow Fraser, who is the only study that we can use as a background for Georgian Bay. They found that all 12 turtles on Beausoleil Island overwintered in 2 large bogs. Although we did not find any of the 8 turtles using peatlands in this study, we know that at least 2 of the males we captured previously in 2019 to the north did use overwintering habitat in peatlands. Use of thicket swamps by Blanding's turtles has not been reported elsewhere, and so this is the first formal documentation, although it appears to be more common knowledge among practitioners. Thicket swamps are widely distributed throughout Tadenac and may therefore be an important habitat feature for blandings and other freshwater turtles. I want to briefly mention the novel monitoring methods that we are including in Tadenac. Besides trapping and using radio telemetry or GPS loggers, we can also use environmental DNA or eDNA. DNA is the genetic material that's shed by turtles, and it can be included in the feces, urine, mucus, sperm, dead skin, hair, and their carcass. This material can be concentrated from the water via filtration and then extracted and then analyzed by qPCR techniques. The benefit is that it is not invasive and does not require any permits or handling of turtles. eDNA can be used to determine that a Blanding's turtle has occupied the wetland or the pond in a recent period within, say, a week's time, depending on weather conditions, and this may reduce the need to do labor-intensive tracking studies. How to sample with eDNA is now the focus of a new master's student, Bryn Hickey. The sampling can involve fairly expensive specialized equipment, such as that shown here, but we've discovered that it can also be collected with fairly inexpensive 2-liter jars that you can purchase at Canadian Tire. And this way of collecting has worked in the winter when the cold weather prevented the frozen equipment from working. This can be a very cost-effective way to collect water by multiple volunteers at different times of the year. Very little training is required, and the filter paper and preservative, which is ethanol, is not expensive. The samples can be kept in the fridge and then passed along to us for later processing at McMaster University. Another novel way to identify turtles is using the unique patterns on turtle plastron and or the spots on the turtle shells. Chantal Markle and I have developed and published this approach for Blanding's turtles, and Bryn is now extending this to spotted turtles. It involves 2 different types of software, one called the I3S Spot for looking at spots on the carapace, and the I3S Pattern for looking at the the pattern of the plastron. We have created a library of the plastron and carapace images of 34 spotted turtles from Ontario wetlands. And using only the test images, we can statistically identify individuals with an accuracy of between 91 and 96%. And so this will eliminate the requirement to notch the carapace of turtles, which is very invasive. We plan to build the library to include another 200 individuals and to build a field app that can be used by practitioners. The preliminary results from last year has already proven the value of studying Blanding's turtles in different regions throughout Georgian Bay. We now have data that can be applied directly to develop effective recovery strategies specifically for Tadenac Bay. And we no longer have to extrapolate from published literature that may not be applicable here. And since we know that other species at risk use similar habitat, such as fox snakes and the spotted turtles, by collecting data on blandings, we are also protecting habitat for many other species. And finally, I want to take this opportunity to share another project that we've just started with 2 Indigenous communities. The project is funded by the New Frontiers in Research Fund, and we are partnering with 2 cottage associations and 2 First Nations to create recovery plans and to develop the long-term monitoring programs for Blanding's turtles in McGregor Bay and the 12 Mile Bay region. I want to acknowledge the financial contributions of the Tadenac Conservation Foundation, as well as in-kind contributions of the Georgian Bay Great Lakes Foundation. A special thanks to Martin Abel, Mark Trudeau, and other Tadenac Club members, and to Gladys Bauer, Colette Isaac, and the Moose Deer Point First Nation members, as well as to logistical support from Mary Muter, Tim, and Sarah Bowman. And I also want to thank all the other graduate students and undergraduate students present and formerly in the Chow-Fraser lab.
Hello everyone, my name is Jacqueline Vinden and I am a master's candidate within Dr. Pat Chow Fraser's lab, and today I'll be going over the long-term changes in E. coli and phosphorus in the nearshore waters of the Georgian Bay Township. The focus of this study is the Township of Georgian Bay, meaning all of the data that were collected between 12 Mile Bay and Severn Sound. As part of this study, we will be using data collected by Dr. Carl Schaefer and his daughter Kim Schaefer between 2001 and 2009. Together, they sampled over 100 sites in various nearshore and inland lake locations, which can be seen on the map on the right here, and they sampled for both total phosphorus, or TP, and E. coli. TP samples were collected throughout the entire period, while E. coli was sampled periodically from 2001 to 2008, with a detailed study conducted in 2009. So in 2009, E. coli samples were collected in 2 to 3 week intervals from early June to early September, with a focus on July and August. As a result of the study, Dr. Schaefer found that the background levels of total phosphorus were below 10 micrograms per liter, which is consistent with the provincial standard for an oligotrophic body of water. He also found that E. coli levels in Georgian Bay were below 10 colony-forming units, or CFUs, of E. coli per 100 milliliters of water, which is well below the provincial water quality objective of 400 CFU. Therefore, he created a new objective called the Georgian Bay Water Quality Objective of 10 CFUs of E. coli. Since Dr. Schaefer's study ended in 2009, no comprehensive study has been done since. This is important since there has been substantive human development within southern Georgian Bay, including the Oak Bay development as well as the proposed developments in Macy Bay since that time. There have also been changes in water levels. Throughout the period that Dr. Schaefer sampled, the water levels were much lower than the long-term mean, but beginning in 2015, water levels rose above the long-term mean, and by 2019, we're approaching record high. So because of this recreational development, as well as the changes in water levels, in 2022, the township wanted an update on conditions just in the nearshore sites. So this doesn't include any of the inland lakes that Dr. Schaefer had sampled in the past. So the township wanted to see what has changed in terms of TP and E. coli in a subset of Dr. Schaefer's sites, and to include additional sites where there has been recent development. Regulatory agencies accept the presence of E. coli as an indicator of fecal contamination and the possibility of pathogenic bacteria, which is a human health concern. So a common test to determine the abundance of E. coli is to incubate a sample within a medium that selects for E. coli, and then that would then fluoresce. So Schaefer and Schaefer designed a volunteer program that uses this method and was carried out between 2001 and 2009 by cottagers. So volunteers filled each well of a coli plate, which is seen on the left here, with water from a specific site and incubated it for 24 hours before they then manually counted the number of fluorescing wells, which can be seen in the right picture. So in 2021 study, we used a different approach because we didn't have nearly as many volunteers, and the Chow-Fraser lab had recently purchased the Tecta B16, which can be seen in the bottom image. So the Tecta can detect E. coli within 2 hours, which is a huge advantage as it provides early warning signs for contamination. Not only is it fast, but the Tecta is easy to use and it reduces the possibility of contamination. However, the basis of the test is the same as the coli plate, where it uses the fluorescence of E. coli in a defined medium to determine the abundance of E. coli, but this is done automatically and doesn't rely on people to physically count the cells, so that helps to reduce subjectivity. Before sampling, we took Schaefer sites and some additional new sites and renumbered them so that each site has a unique identification code. Then during sampling, we standardized water collection across sites by taking water at mid-depth with a Van Dorn sampler. We collected 2 bottles from each site, so there are parallel samples for both total phosphorus and E. coli. We also collected Secchi depth information, as well as used a multi-parameter probe to collect pH, conductivity, temperature, and dissolved oxygen data. Lastly, we measured turbidity and triplicate using the Hatch Porter lab. After we separated the 2021 sites into 5 different regions throughout the township, and going from north to south, they are 12 Mile Bay, Go Home Bay, Cognisheen, Honey Harbor, and Oak Bay, and we included Macy Bay in the Honey Harbor region. So each site was sampled 2 to 5 times between late June and early September in 2021. And the 2021 sites represent about 80% of the sites in the nearshore water that Schaefer had sampled back in 2009. So now moving forward, I'm going to discuss the long-term changes that have been seen from 2009 to 2021, starting with E. coli. So to begin, we did a monthly comparison to reduce some of the variation that occurs throughout the summer. We did a parallel comparison of the 2 years and found that there was no significant differences for the months of June and July. However, we did find that the mean for August of 2009 was significantly higher than the mean for August of 2021. On average, they were 18 colony-forming units of E. coli in 2009 and less than 1 colony-forming unit of E. coli in 2021. However, it is important to note that the methods that were used in each year were different. So in 2009, the coli plate was used, where in 2021, we used the Tecta B16. However, after running some tests, we determined that if the methods were solely responsible for this difference, you would expect to see 2021 having E. coli results that were 2.5 times higher than in 2009, and therefore we can conclude, since this is the opposite, that the methods are not solely driving this change. Next, we wanted to see how E. coli densities changed from region to region. So we took the data from both 2009 and 2021 and grouped them by region and then ran them through a 2-way analysis of variance. To begin, we found that there was no difference between regions. However, we did see that there was a significant decrease in E. coli in 2009 to 2021 in the region of Cognachine. We did see a similar trend with Honey Harbor and Go Home Bay, where we saw a decrease in E. coli from 2009 to 2021, but it wasn't significant. And this is consistent to what we had found before in the month of August. The only region that didn't follow this trend is the region of 12 Mile Bay, where we actually saw an increase in E. coli, but this may be likely to be caused from one very high result in the month of August in Bloody Bay. So what this is telling us is that E. coli densities generally have declined from 2009 to 2021, which may be related to water level changes. And I'm going to go into that further on in the presentation, but it's just something to keep in mind as we're moving forward. So based on those results, generally we can say that conditions seem to be improving. However, that's not the case for all areas, as there are some that are getting worse. So these maps show the hotspots throughout the township in 2009 and 2021. So as you can see, some hotspots have stayed the same. So for example, Monument Channel and Honey Harbor have both been more problematic, both in 2009 and in 2021. But we have also seen that some hotspots have changed. So there have been some new ones that have arisen, like in Bloody Bay as well as in Freddy's Channel. And then there's also been ones that have improved, like in Musquash Channel. It's also important to note that both Macy Bay and Oak Bay were not sampled in 2009, but it's clear now that they definitely are areas of concern. So zooming into Honey Harbor, we can see that the same trend has continued. So again, some hotspots are consistent, like Brandy's Cove, where others have changed. Like Bayview Marina has improved, as well as Sunset Bay, and the channel leading into Honey Harbor as well. So this shows the impact that human development can have, as most of the sites that are still areas of concern are within heavily populated areas of Georgian Bay. So what this tells us is that we should look further into the impact that recreational development is having on water quality, and I will be talking about that further on in the presentation as well. So now I'm going to be talking about the long-term changes in total phosphorus. So this map shows the mean concentration of TP at each site throughout the summer of 2021. But before I delve into any particular data points, I wanted to talk about the TP guidelines put forth by the provincial government. So if a site has less than 10 micrograms per liter of TP, that's consistent with oligotrophic conditions, where if a site has between 10 to 20 micrograms per liter of TP, that would signify mesotrophic conditions. And if a site has more than 20 micrograms of TP, that signifies eutrophic conditions, with 50 micrograms per liter of TP being a trigger point for remedial action. So as a reminder, most lakes in Georgian Bay have a natural background level at or below 10 micrograms per liter, meaning that anything above 15 micrograms per liter, especially anything above 20 micrograms per liter, is definitely an area of concern. So in regards to TP, you can see that 12 Mile Bay, Go Home Bay, and Cognachine do not appear to be an issue, as most of the sites are below 10 micrograms per liter. However, that is not the case for Honey Harbor and Oak Bay, seen in the bottom right of this map, as they tend to have a lot of sites that surpass 15 micrograms per liter, which again is that trigger point. So what this tells us is that we definitely need to further look into Honey Harbor and Oak Bay in terms of TP. So next I wanted to see if there were any significant differences among the regions. And to do this, I grouped them by water level, and that's because there wasn't enough data in each year to have a robust comparison if we group them in years. So just as a reminder, low water levels are between 2001 and 2013, and high water levels are between 2018 and 2021. So I did a two-way analysis of variance and found that there was no significant difference between years, but that there was a significant difference among regions. So we found that Oak Bay was significantly higher than all other regions, while Honey Harbor was significantly higher than Cognachine and Go Home Bay. Secondly, it was found that TP concentrations increased in Oak Bay, Honey Harbor, and 12 Mile Bay. And again, the increase in 12 Mile Bay may be related to the very high TP concentration found in the month of August in Bloody Bay. We also saw that Cognachine and Go Home Bay saw a decrease in TP from 2009 to 2021, which follows the same trend that we saw with E. coli. So what this tells me is that Oak Bay and Honey Harbor seem to be the problem regions for TP and is worth further investigating. So you've heard me mention water levels quite a few times throughout this presentation, especially in how it may be one of the reasons why we are seeing a decrease in both TP and E. coli from 2009 to 2021, and so I'm going to go into that more now. So just as a reminder, throughout the period that Dr. Schaefer sampled, they were sampling when water levels were much lower than the long-term mean, which is seen in the green line on this graph. However, when I was sampling in 2021, we were sampling when the water levels were much greater than the long-term mean. So what this means is that when you have higher water levels, there's a greater volume of water within the bay, which can lead to more dilution of nutrients and solids. However, when you have lower water levels, you have less volume, which can lead to an increase in concentration of those nutrients and solids. So what this means is that if you had the same amount of nutrients within both scenarios, you're more likely to capture those nutrients within your grab sample when you have low water levels, which would make it appear like the water level is worse, than during high water levels, where you're less likely to get them in your grab sample, which may make it seem like the water quality has improved. So this trend is captured in the mean TP profiles of two sites that were sampled by the Severn Sound Environmental Association from 2003 to 2021. So when the water levels were lower, you could see that the nutrient concentrations were increasing, but when the water levels began to rise again, we could see that the nutrient concentrations began to decrease. So it's important to note that we are not contributing this every change that we saw entirely to this effect. We're just saying that this generally is a trend that we've been seeing, since there are definitely some very specific site factors that can be influencing water quality as well. And some of those site factors are what I'm going to be going over now. So one of the factors that may be impacting water quality at the Macy Bay wetland, which is seen in the top left picture, is that it's located very close to a sewage lagoon, only 200 meters from one, and that might be why we're seeing very high E. coli and total phosphorus levels there. Another factor that might be impacting water quality in the Oak Bay wetland is the fact that it's situated right beside a golf course. And they have increased fertilizers and nutrients going onto the golf course that may be entering into the water, leading to increased E. coli and TP concentrations. Then lastly, being situated within areas of high recreational development, like within marinas and areas with a high amount of cottage density, like Bloody Bay, Church Bay, and Brandy's Cove, may be one of the reasons why they're seeing increased levels of E. coli and TP. So the trend of being situated within areas of high recreational development leading to higher TP can be seen in this data here as well. So we had a volunteer that collected TP data from 10 different locations around the Brandy's Cove area throughout the summer. And if we're looking specifically at the July 11th data that was collected, we can see that sites 1, 2, 3, and 7 had the higher amounts of TP, and those are the sites that are located right around the marina, whereas sites 5 and 6 had lower levels of TP, and they were situated more in the open water. This trend is also observed when looking at the average TP and E. coli data that I had collected in the summer. So when looking at Site 101, again, it had the higher levels of TP and E. coli, and it was situated right near the marina, where sites 1049 and 1053 had lower levels of E. coli and TP, and those were the sites that were situated closer to the open water. So based on what we have seen so far, I wanted to create a summary of my observations. So the first summary is that Honey Harbor and Oak Bay tend to have higher levels of TP and E. coli, and this may be due to the fact that they are in regions that have higher levels of recreational development. The second factor that I observed is that land use seems to be a key characteristic in determining water quality. And so when looking at land use specifically in regards to recreation and recreational development, we are seeing that the regions that have increased recreational development tend to have poor water quality. In regards to recreational development, there are two main factors that we will be looking at. The first one is in regards to boating anchorages in marinas. And so typically these are within enclosed bays with low flow exchange rates, which can lead to a buildup of pollutants, which includes fecal bacteria. And then this can be worsened by the introduction of fecal waste from liveaboard boats through leaking holding tanks or the illegal dumping of black water. And then the second factor that is part of recreational development is in regards to cottage density. So we're seeing that increased levels of shoreline development can introduce fertilizers, sediments, and sewage effluent into the water, which can disrupt natural nutrient cycling and leading to increased levels of TP and E. coli as well. So lastly, we are seeing that temporal changes don't appear to be as strong a factor. So even though water levels did seem to play a role in some regions, specifically like Cognachine and Go Home Bay, we're seeing that they aren't playing as big a role as those land use factors are. And so what this tells us is that we need to look into metrics of human disturbance as well as some other influences that may be impacting water quality, potentially like rainfall intensity. So based on the results from the past year, we have decided to focus our sampling within Honey Harbor and Oak Bay region, since those are the regions that had the worst or the highest levels of nutrients as well as E. coli. So we'll be sampling there 5 times for both TP and E. coli throughout the summer, while also sampling in the problematic areas in Go Home Bay and 12 Mile Bay at least once in the summer to see how those have changed over the years. Before finishing, I just wanted to say a huge thank you to various members of the community for their support of this project. So this includes Jean DeMarco, Mary Muter, and David and Brenda Flower for hosting us this past summer, as well as for taking me out sampling to various locations. I also wanted to thank the Township of Georgian Bay, McMaster University, as well as the Natural Sciences and Engineering Research Council for their financial support. Thank you.
It looks like people are shy about asking questions because we're certainly not getting any. I don't know the reason for that, but in any case, we're happy to answer questions if anybody wants to post anything now. I just want to thank all of the speakers. This was excellent. Huge amounts of knowledge shared. It is disappointing the number of attendees that we've had, which seems low, but when I add them all up, it's probably around the same number that we had in person the last time we were able to gather at the McMichael Gallery. So I just thought via the internet that more people might join. I was pleased to note that yesterday a number of Americans from Michigan did listen into our webinar. Because nothing is going to happen about water levels if we don't get support from Americans on Lake Michigan and on the US side of Lake Huron. I just once again want to thank everybody who has joined us and to our speakers tonight. I would like to recognize that Roy Schatz is no longer with us. He sadly passed away due to cancer last fall. He has been our guiding light through this work for over 20 years. prior to that, he was a past president of the Georgian Bay Association, and he was the founding president of GBA Foundation. And then he has stayed with us. There was no one that could translate complex science into language that the average person could understand as well as Roy could. We miss him greatly, but we are trying to move on. We have 2 new board members, Adam Scott from Go Home, basically a climatologist, so providing us expert advice on climate change. And Bruce Rogers from the cottage down by Meaford, another hydraulic engineer to add to our team for that work. As Jeff has pointed out, we are posting the webinars on our website. And on our website, you can figure out by clicking on donate how to donate. It's pretty obvious from this work that our funds are put to good use. We do not have any full-time staff. Literally, nearly all the funds that are raised are used for this research. I do want to thank Jeff Simpson, who has orchestrated this webinar for us. He's been great to work with, and we appreciate his help so much. Unless any of the other participants or the presenters have comments.
I just wanted to thank you for the opportunity to share the data that we've been collecting over the last couple of years, that we've not had a chance to share this very, very important research. And I also want to thank all the people that have also helped us that we couldn't name in the acknowledgements, but there've been a lot of cottages over the last 20 years that have helped us.
Yes, thank you, Pat. That is so true. So hopefully, maybe even late next fall, we might be able to gather in person, provided COVID numbers don't go back up again in the fall. And I think everybody is hoping that that will not happen again. So again, thanks to everyone, and we'll see you online. You can send emails anytime to us via info@georgianbaygreatlakesfoundation.com, or through our website, you can get information and figure out how to donate. So thank you to everyone, and hope everyone has a great summer.
Bye-bye.
Bye for now.